Oh, there we go. We're recording. I will expect you to know all those stages of mitosis, be able to put them in order, and be able to recognize them. One type of test question I have been known to throw is putting pictures of a nucleus undergoing a stage of mitosis and asking you what it is. So make sure you know those. Make sure you know those pretty well. Let's see. Let's go on back to where we were. And there are our slides. So let us keep marching onwards. And here's where we have to do some counting. And this gets a little bit tricky for some students to keep track of. When we start mitosis and the chromosomes become visible, we can see them, we can count them. And at the start of mitosis, each chromosome in a cell consists of a pair of chromatids. Remember, those are those two strands that are connected at a point called the centromere. When mitosis is done, each chromosome consists of only one chromatid. The chromatids separated when anaphase started and the centromeres broke apart. But you could watch the daughter cells, that's the two cells produced by mitosis, and you could watch them until they divide again. And each daughter cell will start mitosis with two chromatids per chromosome. So somewhere during interphase, somewhere where you can't directly see it, each chromosome sprouts a new chromatid somehow. So there's something going on in interphase that we can't immediately see. And we now talk about cells going through what we call the cell cycle. Mitosis, which is abbreviated M here, actually might not take up that much of a cell's lifespan. Again, it might take an hour, but the rest of a cell's lifespan might take a whole day, maybe until the next cell division. And we can divide interphase into sub-stages that we call G1, S, and G2. G1 is called the first growth stage. And this is the cell just growing and metabolizing and doing whatever it's doing and talking on its cell phone. Already used that joke, sorry. And then somewhere in interphase, it goes through the S phase where the cell creates new chromatids. We can't directly see that happening, although there were some really neat experiments in the 50s that were able to demonstrate when it happens. And then after the S phase, we have a stage called G2, the second growth phase, where the cell might grow some more and then prepares for going into mitosis. And exactly how long these take depend on what cell you've got. There are some cells like mature nerve cells, for example. Mature nerve cells pretty much get out of the cell cycle and are permanently stuck in G1 somewhere. But there are cells in the brain that are not stuck and they can develop into new nerve cells. We actually talked about those way at the beginning. But yeah, the actual timing depends on what cell you've got and what species it is. But by and large, this is what's going on in interphase. There has to be some stage in there where the chromosomes sprout new chromatids, even if you can't see it happening. That means we can divide interphase into G1, S, and G2. Why the hell does this matter? Well, mitosis is the way that single-celled eukaryotes reproduce. So all of your single-celled protists reproduce by mitosis. It's also the way that development happens. And when we're talking about development, we mean development from a fertilized egg to an adult. You started out as a single fertilized egg cell. You currently consist of roughly 37 trillion cells, give or take a few. And with some limited exceptions, all of them came into existence through mitosis. When that one fertilized egg divided into two, and the two divided into four, and the four divided into eight, dot, 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 all the way to adulthood, that's all there because of mitosis, with one limited exception we'll get to in a minute. Mitosis is also responsible for physical growth and maintenance. You lose 50 to 70 million cells in your body every day. The cells that make up the cornea of your eye, the part that you would stick a contact lens on if you had one, you lose those. Their typical lifespan of a mature corneal cell is roughly 1 hour. They're constantly dying and breaking off and getting washed away in your tears. The skin cells on the outside of your body are, the outermost layer is actually already dead, and those cells are constantly breaking up and flaking off. On your scalp, we call that dandruff, but it happens on the rest of you as well. And those have to be replaced from below by new cells. You shed millions of cells from the lining of your intestine every day. Those just slough off and pass out in feces, and they have to be replaced, and so on. Wound repair is mitosis. If you've ever gotten a cut and it's healed, that's done through mitosis, growing new tissue. And then finally, we probably won't have time to get into exactly how this works, but your cells have various controls over the process of mitosis. It doesn't just happen automatically. There has to be a certain set of events that go on that trigger the cell to enter the mitosis pathway. If those controls get damaged, one result is cancer. Cancer is fundamentally mitosis out of control. It's a lump of cells that are all dividing as fast as they can and can't stop. Just like a car might have faulty brakes and a stuck accelerator, cancer cells have a stuck accelerator and faulty brakes on the little process that they undergo to trigger mitosis. They're growing, they can't stop. They can form a big lump. That's what a tumor is, just a lump of cells. They drain your body's energy, and they can spread. Yeah, I won't go too much into what cancer is, except that at its base, it is mitosis out of control. So understanding mitosis is critical for a lot of reasons. But it gets worse because mitosis is not the only type of cell division that we see. There's another type called meiosis, M-E-I-O-S-I-S, meiosis. Eurosis? No, meiosis. And mitosis, which we've seen, is responsible for the existence of almost all of your cells. Almost every cell in your body ultimately came about through mitosis, going back to the very first fertilized egg cell that you came from roughly 21 odd years ago in most of your cases. Meiosis is the way that you produce egg cells or sperm cells, whichever the case might be. In very medically very rare instances, it could actually be both. At least in humans, it's very rare. There's plenty of organisms that do produce both, but we don't need to get into that. Meiosis is how you make eggs or sperm. And to understand it, we have to give you some background on chromosomes, because people started looking more carefully at the chromosomes that they saw. And it turns out that when you look at them, they're not identical. Some chromosomes in a cell will be long, some will be short, some have the centromere close to the physical center, and some the centromere is kind of off-center. We call those centric and acentric chromosomes. And if you use special staining techniques, chromosomes have stripes on them. They have bands. You didn't see those in the slides that we just looked at, but with special staining techniques, you can spot the chromosomal bands. And it turns out that if you take a cell that's dividing and you sort the chromosomes, and at least in the old days, you would physically do this by taking pictures and then cutting out individual chromosomes and sorting them. It turns out that in a typical eukaryotic cell, there are 2 of each type of chromosome. And a cell that has 2 of each type is said to be diploid. Not deployed, as in, you know, sent off to fight a war somewhere. Diploid. Diplo happens to be Greek for double. So a diploid cell has double something. So here's what this looks like. These are some chromosomes from a normal human female. They've been stained with both a red and a yellow, or bright yellow green. They've been stained with both a red and a yellow fluorescent stain. And what you can see is that some of them are almost all red, some are almost all yellow, and a number of them have patterns of stripes or zones of red and yellow on them. So number 7 has some nice yellow striping on it, for example. What I want you to see here is, first of all, you can see the 2 chromatids joined at the centromere very clearly in this case. And second, there are 46 chromosomes, and you can sort them, and you can match them up by size, sorting from largest to smallest. You can sort them by size and centromere position. So the centromere for chromosome 1 is very close to the center. It's a centric chromosome. But the centromere for chromosome 13 is right there at the edge. That one's acentric. And you can match them up by banding pattern. And what you get is 23 pairs, and they're numbered 1 through 22, with the last pair numbered X. Those are the sex chromosomes, and we'll get to those later, probably not till Friday. But for the moment, just know that we've got 23 pairs of chromosomes, sorted by size and centromere location and banding pattern. Different animal and plant species have different chromosome numbers. These are mouse chromosomes that you're seeing here. They've been stained in a different way, so the bands look a little different. And mice have 40 chromosomes, and those make up 20 pairs per cell. There's no simple relationship between how complex an organism is and how many chromosomes per cell it's got. It's just a number. Mice have 40, fruit flies have 8. You've got 46, horses have, I think it's 66, or it might be 64, I forget. The record is this tiny little plant called an adder's tongue fern that you could step over and never even notice, that has 1,260 chromosomes per cell. On the other hand, there are some animals that only have 2. And it's just a number. There's no simple relationship between how many chromosomes per cell an organism has and how complex or advanced, quotation marks, it might be. It's a number. All it is a number. Animals and plants and many eukaryotes form specialized cells for sexual reproduction called gametes. And in the cases you're familiar with, gametes come in 2 forms, large and small. The larger gamete is called an egg, and the smaller gamete is called the sperm. Typically, an organism that makes eggs makes fewer of them than sperm, but on the other hand, sperm are considerably smaller than eggs. This applies to plants. The eggs are buried deep down in the flower, and the sperm are contained in pollen grains. This is true for animals. In many protists and fungi, the gametes are all the same size. We might call these plus type and minus type, or type A and type alpha, or something like that. But we don't call them eggs and sperm because the gametes look identical. That's true of a lot of seaweeds. In fungi, instead of separate gametes, you have two hyphae that will fuse, and the hyphae look identical, but there are markers on them that the fungus can tell, but we can't. So there are some critters where the gametes all look the same, but certainly in animals and plants, you have large ones and small ones. And the thing is that gametes only have one of each pair of chromosomes, and we call this being haploid. Right? If you've got two of each pair of chromosomes, it's called being diploid. If you have one, it's called being haploid. We're not really going to need to worry about when this happens, but it is possible for some cells to have three of each chromosome, which would make them triploid, or four of each chromosome, which would make them tetraploid, or five of each, which is pentaploid, and so on. For our purposes, we're not really going to need to worry about how that can happen. Meiosis is the process of cell division that produces haploid cells from diploid cells. Another name for meiosis is reduction division, because unlike mitosis, meiosis has to cut the number of chromosomes per cell in half. If you have 46 chromosomes in most of your cells, your gametes, your eggs or sperm, whichever it is, your gametes must have 23, being half of 46, duh. In fact, specifically, if the cells in your body mostly have 46 chromosomes per cell, organized in 23 pairs, then the purpose of meiosis is to give you eggs or sperm that have 23 chromosomes consisting of one of each pair of chromosomes. So you don't just have 23 chromosomes, you have one of your chromosome 1 and one of chromosome 2 and one chromosome 3, ... all the way down to the end, in every one of your eggs or your sperm. The basics of how this works was worked out in 1883 by a guy named Edward van Beneden, who, among other things, studied those intestinal roundworms whose cells we looked at in the last lecture. In animals, meiosis is how you make eggs and sperm. With plants, things get a little weird, and I probably won't have time to talk about this. Plants can be a little bit strange about when meiosis happens. And yeah, we probably don't have time to get into that. But in animals, this is how you make eggs and sperm. So that's a human egg taken with a gizmo called a scanning electron microscope, caught in the act of being swarmed with a whole bunch of human sperm cells. There might be 100 million sperm cells in each ejaculation, but only a few hundred might actually make it to the egg cell because they refuse to stop and ask directions. Actually, I'm kidding. That was a joke. But the egg cell has one of each pair of human chromosomes, 23 each. Every sperm cell, if it's normal, has one of each pair of human chromosomes, which is 23 each. And so when Mrs. Egg and Mr. Sperm fuse together, they form a cell known as a zygote. That is a zygote right there. If you look in the center, you can actually see two nuclei that are in the process of fusing right there. And the zygote, the fertilized egg, now has two of each chromosome, 23 plus 23, which is 46. That zygote can then divide by mitosis into 2 cells, 4 cells, 18 cells, 8 cells, 16 cells, 32 cells, dot, 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 and ultimately give rise to a fully formed adult. If there's a mistake in meiosis, an egg or a sperm can end up with 2 copies of one chromosome or possibly be missing a chromosome. There you've got some human chromosomes that have been stained. They've been stained with a different technique from that fluorescent stain that we saw earlier. But you can see that there is one pair of chromosome 1. And if you look at that pair of chromosome 1, one of those chromosomes came from mom and one of those chromosomes came from dad. We can't tell which, and it's not important here. If you look at chromosome pair 2, one of those chromosomes came from mom and one came from dad. Chromosome 3, one of those came from mom and one of those came from dad. And so on until we get down to chromosome 21. And if you look carefully, there are not 2 copies of chromosome 21. There are 3 copies of chromosome 21. That is because either the egg cell or the sperm cell, doesn't really matter which, carried 2 copies of chromosome 21, and then that third was added by the other gamete. So one of the gametes had 1 copy of chromosome 21, and the other one had 2 because meiosis made a mistake. This, by the way, causes a genetic condition called Down syndrome. People with an extra copy of chromosome 21 have some physical features, tendency to an enlarged tongue and epicanthian folds on the eyes, tendency to some internal features. They can be prone to heart and intestinal problems, and generally lowered intelligence. I'm assuming you've heard of Down syndrome before. This is ultimately why it is. 3 copies of chromosome 21 are present. It can happen, by the way, to other chromosomes, but usually that results in a condition that, as they say, is not compatible with life. A zygote that just happened to get 2 copies of chromosome 1 from one parent plus a third from the other parent would probably end up miscarrying, quite possibly before the woman is even aware that she's pregnant. You will occasionally get babies born with 3 copies of chromosome 14 or 3 copies of chromosome 18. Usually, those babies don't live very long. There's a few that manage to defy the odds, but most of the time, if you've got 3 copies of 14 or 3 copies of 18, you'll die within a few days after birth. Although some of them manage to hang on, but it's pretty rocky. They don't thrive most of the time. There's those. There, computer animation. I'm making the red square blink. There. So meiosis is how you produce haploid cells from diploid cells. And I'm not going to make you learn all of the stages of meiosis. You're welcome. What you need to know is that meiosis is actually a double division. It's divided into meiosis 1 and meiosis 2. At the end of meiosis 2, you have not 1 cell, not 2 cells, but 4. At least if you're making sperm. Egg cells, it's a little bit different, and I probably won't have time to get into how it is that with egg cells you don't produce 4 eggs, you produce 1 egg and a couple of little lumps called polar bodies. Just know that meiosis to make eggs is a little bit different from this, but we don't need to go into what the difference is. Before meiosis, a cell goes through those G and S phases. So when we start, the cell has 2 of each chromosome, and each chromosome consists of 2 chromatids. In meiosis 1, 1 of each pair of chromosomes goes to each daughter cell. So at the end of meiosis 1, you have 2 cells, each with one of each chromosome. And then at meiosis 2, it's just like mitosis. Each chromosome splits and one chromatid goes to each daughter cell. So keep track of this. Before meiosis, we have one cell. That cell has two of each chromosome, and each chromosome consists of two chromatids. At the end of meiosis 1, we have two cells. Each cell contains one of each chromosome, and each chromosome consists of two chromatids. And then at the end of meiosis 2, we have four cells. Each cell contains one of each chromosome, and each chromosome consists of one chromatid. That's what you need to absolutely know, plus a couple of other things I'll show you in just a minute. But here's what this looks like. Meiosis 1, these are cells from lily anthers. Anthers are the organs that produce pollen grains. So what you're looking at here is developing cells that will end up becoming plant sperm. And they're all undergoing meiosis. And meiosis 1 looks like mitosis. And it starts with prophase 1, in which the chromosomes condense. So on the left, you've got early prophase, where the chromosomes are still looking very narrow and long and stringy. They're shorter and thicker in late prophase 1 on the right. Something else happens, and you need to know this. This is a critical difference between mitosis and meiosis. At the beginning of meiosis 1, but not mitosis 1, or not mitosis, but in meiosis 1, each chromosome pairs up with the other member of its pair. So the two copies of chromosome 1 pair up and get together, and the two copies of chromosome 2 pair up and get together, and they form structures. They're called meiotic tetrads. That's what you're seeing here. These funny things that look like X's or loops, that's actually pairs of chromosomes that are stuck on each other. And the paired chromosomes undergo something called crossover. They exchange equivalent pieces of each chromatid with each other. So when the two chromosomes are paired up, they'll both break and swap pieces. They'll both break at the same point and trade pieces, or each one will pick up the other one's broken piece. That's called crossover. And there you've got an actual picture of it over there on at the top, with a diagram of what's going on over there on the right, where those chromosomes are crossing over. It makes a little X-shaped structure that's called a chiasma. Don't worry about that term, but that's what crossing over looks like, and it's something that happens only in meiosis, not in mitosis. Meiosis 1 ends with the formation of two haploid cells. So it looks like there we've got anaphase 1. One of each chromosome pair will move to each daughter cell. And then meiosis 2 is exactly like a repeat of mitosis. The chromosomes line up, they separate at the centromeres, and each chromatid moves to opposite ends of the cell. So that's what meiosis 2 looks like. And here's the end result. We end up with 4 cells that are haploid. So once again, we started with one cell that was diploid. It had 2 of each chromosome, and each chromosome consisted of two chromatids. One cell, 2 chromosomes, 2 chromatids per chromosome. At the end of meiosis 1, we had 2 cells with 1 of each chromosome, and each chromosome consisted of 2 chromatids. At the end of meiosis 2, we have 4 cells each of which has only one of each chromosome, and each chromosome consists of only one chromatid. So each of these cells has only one of each chromosome, but each of these cells is going to become a plant sperm cell that will, if all goes well, find a plant egg cell that also has one of each. And when those two cells get together and decide that they like each other very much and are willing to make a commitment, they join together, they fuse their nuclei, the two nuclei become one, and the resulting zygote will have two of each chromosome again. Why am I hammering on this? Well, think. Mendel worked out, just by crossbreeding an ungodly number of pea plants and counting them, he worked out that genes come in pairs, right? You normally have two of each gene, and of course, each copy of that gene can be one or another allele, right? Yes. You've got two copies of each gene, and you pass on one of each pair to each of your offspring. So if you have, that cat that was annoying the heck out of me at the beginning of lecture is heterozygous for the white spotted gene. His genotype would be big S, little s, because he's got one copy of the spots gene and one copy of the solid gene, or the solid allele, right? He's not going to have any kids because we took care of that surgically a long time ago, but if he did, he would pass on either his big S allele for white spots or his little s allele for solid color, but he wouldn't pass on both, right? That's the basics of Mendel's laws. Now here we've got a bunch of microscopists that have worked out what happens in cell division by about the year 1880, and all of a sudden they've noticed that, when you produce gametes, each gamete contains one of each chromosome pair, so you'll pass on one of each of your chromosomes, one of each pair of chromosomes that you've got, you'll pass on to each of your offspring. And you got one of each of your chromosomes from each of your parents. You got one copy of your chromosome number 1 from mom and the other copy from dad. So this is why this matters, is that we've been working on a jigsaw puzzle from two different directions, and it looks like that chromosomes are following the same basic rules as Mendel's genes. Mendel never got to see this. I don't think this wasn't worked out in his day, or at least it wasn't until he had to give up science and become the abbot of his monastery, poor man. But it turns out that chromosomes and genes following the same rules was evidence that chromosomes and genes have something to do with each other. Now, we know that chromosomes are not genes because, for example, pea plants have, I think, 7 pairs of chromosomes, but a heck of a lot more than 7 genes. Humans have 46 chromosomes, but I can't even remember how many genes we've got. I forget the exact number. It's something on the order of 20,000. So a chromosome isn't a gene, but chromosomes are the physical basis for what genes are. We would now say that genes are carried on chromosomes, and I'll talk about that in more detail coming up on Friday. But the reason why you've got to go through all this is that these are two ends of the same jigsaw puzzle, and we're in a position where we can connect the two and start going from there. So, right. With that said, I'm going to go ahead and stop the recording and call it a day, or as my wife's family would say, I'm going to bless this and call it dinner.